Alrighty, so good morning, good afternoon, and good evening, everyone, and welcome to the Global Virus Network's Frontiers in Virology webinar. And today I have the great pleasure to introduce our speaker. Dr. Aditya Bade is an associate professor in the Department of Pharmacology and Experimental Neuroscience at the University of Nebraska Medical Center. Dr. Bade's research primarily focuses on neurodevelopmental pharmacology and toxicology. His current work is focused on identifying underlying mechanisms of antiretroviral drugs and HIV comorbidities linked to adverse pregnancy outcomes and developing intervention strategies. Notably, he was the first to demonstrate how dolutegravir inhibits matrix metalloproteinases, impacting fetal neurodevelopment and processes long-acting injectables as a preventative measure against antiretroviral linked neurodevelopmental impairments. With 39 peer-reviewed publications in esteemed journals, Dr. Bade maintains highly productive research profiles and is focused on neuro-health and disorders. His work has been supported by multiple intra and extramural grants. Overall, Dr. Bade's overarching goal is to improve health outcomes of HIV-1 positive mothers and their fetuses. So please join me in welcoming Dr. Bade and his extremely important work in the current work that he's going to present, which is Developmental Deficits in Offspring of HIV-1 Mothers, Bridging the Known and the Unknown. Dr. Bade, you are welcome to take it away.
Thank you, Elizabeth, for the kind introduction. So let me share the screen. Uh, is it visible?
Yes, we can
Yes,
see.
looks good.
Okay, perfect. Just give me a second. Good morning, everyone. First of all, thank you. I would like to thank GVN organization and committee members for giving me the opportunity to present my research at this international platform. Also, additionally, thank you everyone for actually joining the seminar. I know it's quite early morning for few of you who might be located at the West Coast. So today, from my presentation in next 45 to 50 minutes, my objective is to provide the summary of research work completed in my laboratory, as Elizabeth mentioned, which is focused on the developmental neurobiology in the field of HIV research, and what the future looks like in coming years for us. Additionally, I have added a little bit extra background so it can remain an educational material for the early stage investigators. So here is an figure which provides a overview of the research questions we are trying to address. So till now, several clinical as well as preclinical studies have shown the increased risk of neurodevelopmental impairments following gestational exposure to antiretroviral drugs. And this could happen through direct exposure of central nervous system to antiretroviral drugs, or even through the indirect mechanisms in which antiretrovirals could influence placental development or maternal mental health. And as both of these biological factors are known to play a critical role in determining pregnancy outcomes, as well as deciding or contributing in deciding the neurodevelopmental health as well. However, addressing these interactions is not that simple. Yes, antiretrovirals could have direct impact on fetal central nervous system, but at the same time, majority of people living with HIV struggle with some sort of substance use disorders, which also needs to be considered. Additionally, due to the benefits of antiretroviral drugs, majority of these children remain HIV-free. However, even the exposure to the viral proteins or sustained low level of inflammation in maternal system even can affect the pregnancy outcomes. So basically, these are the questions we are trying to address, very complex interactions to study, particularly in the basic science area, where we currently lack appropriate research models to really study the maternal fetal HIV interactions. So we are trying to address one question at a time, developing models which can help us to address these questions. And I'm going to provide a snapshot of some of the discoveries we have performed. So before I begin, I would like to thank and introduce my lab members to whom I'm very proud to mentor. Basically, they are the ones who really work hard day in, day out with the dedication, and I'm basically going to present the summary of their hard work. So as I earlier mentioned that my research focus is majorly on the developmental neurobiology, currently in the context of toxicities associated with antiretroviral drugs. So my laboratory's research is particularly based on three major pillars, which are identification of biomarkers or underlying mechanisms associated with the neurodevelopmental impairments following gestational exposure to ARVs, developing novel in vivo bioimaging tools for early detection of these developmental deficits, as well as developing therapeutic intervention strategies to improve the safety as well as therapeutic efficacy of broadly utilized highly potent antiretroviral drugs. And today, I will try to present a little bit on each of these areas, and hopefully, I will have enough time to do so. So when we talk about the antiretroviral drugs or neurodevelopmental impairments in the field of HIV, maybe it's better to have a little bit perspective from the history as well. So as all of us know that it has been more than 40 years since the first patient with HIV was reported, which was in early 1980s. And since then, remarkable progress has been made with the development of highly potent antiretroviral drugs, not only for the treatment, but also for the prevention strategies. So when the patient is adhered to these antiretroviral drugs, he or she can expect a almost a normal life as like a normal uninfected person. However, despite such advances seen over the last 40 years, one special population that includes the uh, pregnant woman living with HIV and its fetus remain vulnerable to the toxicities, particularly with the antiretroviral drugs. And till today, we still don't really know this particular or specific effects of broadly utilized antiretroviral drugs on the special population, and thus obviously the intervention strategies are not there. So if we quickly look at the current stats of HIV positive people, just to know how big of the public health crisis it is, currently around 39 million adults are living with HIV infection, and out of those 39 million, 45% are women of reproductive age, which comes around 15 to 16 million population. And currently pregnant women with HIV are recommended to maintain the antiretroviral therapy throughout pregnancy and even during the breastfeeding period, particularly in resource-limited countries. And this is particularly due to the benefits of antiretrovirals in preventing the vertical transmission of virus from mother to its fetus. Without antiretroviral therapy, the transmission rate used to be around 40 to 50%, whereas antiretrovirals have significantly reduced it down to less than 1%. And with the improved availability of these antiretroviral drugs at affordable cost worldwide, more than 1 million this pregnant women with HIV currently, more than 80 to 85% of people, pregnant women living with HIV, have access to these antiretroviral drugs, and thus each year more than 1 million infants are born to these pregnant women who remain uninfected. So, however, along with such benefits, one risk was introduced, which was exposure of these fetuses to the antiretroviral drugs, particularly during the developmental period. And we all know that which remains vulnerable or susceptible to the external modulators such as antiretroviral drugs. And several clinical and preclinical studies have showed that the HIV children, HIV-exposed uninfected children, have poorer neurodevelopmental outcomes compared to the HIV-unexposed uninfected children. And these neurodevelopmental impairments have occurred across multiple functional domains. These include, but not really limited to, language, gross motor cognition, or socio-emotional outcomes. These children are also at higher risk of preterm delivery, immune abnormalities, infectious morbidities, or even impaired growth. And these factors are also known to affect the neurodevelopmental outcomes. So, with the acknowledgement of such a large population getting exposed to antiretroviral drugs worldwide, and this number is expected to increase in coming years. So it is very critical to make sure that the pregnant woman and his fetus not only receive the care they deserve, but without any adverse events. So when we talk about the antiretroviral drugs, till now more than 30 antiretrovirals have been approved by US FDA, and they basically are classified into 9 different categories based on at what stage of HIV cycle they act on. And their recommendation for a pregnant woman depends on like whether it's initiated during pregnancy, prior to pregnancy, where the pregnant woman is naive or experienced to these antiretroviral drugs, resistance profile. I mean, there are multiple factors. However, here is a list of drugs which is recommended for a pregnant woman who is naive to antiretroviral drugs. And the drugs which are highlighted in green are currently preferred. The blue ones are the alternatives and red ones are currently not recommended. So, out of all these antiretrovirals, today I'll be majorly focused on one class, which is integrase transfer inhibitor because of their broad utilization to treat the adult as well as pregnant women population. And especially I'll be talking about dolutegravir because of its broad utilization worldwide and known clinical adverse events. So dolutegravir is currently recommended as a part of first-line and second-line regimen due to its properties, and special mentions include high potency, limited drug-drug interactions, and most importantly, high genetic barrier to viral drug resistance. So due to these properties, when the DTG came out of patent around 2015, in summer of 2017, several organizations came together such as Melinda Gates Foundation, Clinton Health Initiatives, World Health Organization, United Nations of HIV and AIDS, and many others, and signed an unprecedented agreement to increase the availability of this DTG-based generic regimen, which is called STLD, at the price of $75 per person per year in resource-limited countries. And currently, I even read that in some of the countries, it is even in less than $50 per person per year. And due to this pricing agreement, more than 100 resource-limited countries have incorporated this DTG-based generic regimen into their national guidelines of treatment. Around 2018, almost 3.9 million people were on this regimen. 2019, 6.9 million people. And currently, it is believed that almost half of the world's total HIV population might be getting treated with this DTG-based regimen, as majority of HIV population resides in resource-limited countries. And when we talk about the resource-limited countries, women of childbearing age remain a significant HIV-positive population. So when we just look at the new infection data, in worldwide, almost 50% of the new infections occur in women, and every week, 4,000 young women aged between the 15 to 24 years become infected with HIV. However, when we talk about sub-Saharan Africa, which currently holds 2/3 of the world's total HIV population, 63% of the new infections occurred in only in women, and out of 4,000 young women who became infected with HIV worldwide every week, 3,300 new infections occurred only in sub-Saharan Africa, which comes around 80% of the world's new infection population. So with the increased availability and incorporation of this DTG-based regimen into national treatment guidelines, as well as increasing pre-treatment resistance to NNRTIs such as efavirenz, majority of these pregnant women or women of reproductive age living with HIV are and will be treated with DTG-based regimen. So one of the major concern which was raised in the actually summer of 2018, wherein during that time, a observational study called SEPMO study from Botswana reported that there is an increased risk of neural-tube defects in infants who were exposed to, who were born to mothers taking dolutegravir at the time of conception. In the beginning, the risk was reported as 8 to 9 times higher than any other antiretroviral drugs taken at the time of conception. Later, additional data were reported following summer, and the risk was much lower, but it was still reported as 2 to 3 times higher. And currently, with the additional data, it is reported that the risk is similar as any other antiretroviral drugs at the time of conception. Another study called SMART Code study, which was conducted in North America, It also evaluated the associations between the gestational ARV exposures and postnatal neurological outcomes. Even though with the very limited sample size, the study showed a potential of DTG exposure with the adverse neurological outcomes, and particularly when the exposure is happened during the first trimester or at the time of conception. I mean, these are just the couple of studies. Obviously, we still lack the more detailed assessment of how these broadly utilized drugs affect the neurodevelopment in this particular spatial population. And in addition to these clinical studies, there were multiple preclinical studies as well, which have shown the potential risk of neurodevelopmental impairments associated with the dolutegravir. And in addition to the direct effects on fetal outcomes, adults who are also getting treated with the DTG-based regimen are known to be at higher risk, particularly women at higher risk of neuropsychiatric adverse events or other metabolic disorders such as weight gain, diabetes mellitus, or hypertension. And if these comorbidities also occur during pregnancy, could have adverse impact on neurodevelopmental outcomes. So based on all of this, what's our major question that we tried to answer was whether and how DTG and other integrase transfer inhibitors affects neurodevelopment, whether do they have short or long-term effect on neurodevelopmental outcomes, and what could be the underlying mechanisms. Obviously, due to initial just to give a little bit background, like obviously due to the initial association with the neural tube defect, significant efforts were employed by the clinical as well as preclinical researchers, just tried to figure out the association between the DTG and folate levels. People tried to look at the effect on maternal plasma folate levels, RBC folate levels, or folate transport pathways, folate receptors, amount of folate reaching to the fetuses. However, no study could find a solid association between the DTG or cause and effect relationship between the DTG and folate deficiency. So when we try to understand the mechanism through which DTG could impact the neurodevelopment, we realize that the DTG possesses a metal binding core or metal binding pharmacocore in its chemical structure, through which it can basically possesses a potential to interact with some of the metalloenzymes required for normal biological activities. And out of all metalloenzymes, we were particularly interested zinc-dependent proteases known as matrix metalloproteinases because of their critical role during neurodevelopment. In general, MMPs are described in the context of extracellular matrix degradation or remodeling. But however, now it is very well shown or established in the field that the MMPs are expressed at the higher concentrations or higher levels, during early neurodevelopment. And they play a critical role in regulating several cellular processes required for the CNS development, such as neurogenesis, axonal guidance, outgrowth, synaptogenesis or plasticity, etc. As MMPs are very potent proteases, their activity is tightly regulated. So whenever there is an imbalance in their functions or activities, whether it's increase or decrease, it could have a detrimental impact on central nervous system. So to begin with, we came up with the working hypothesis that the DTG inhibits the MMP's activities. So to test our hypothesis, we perform a gold standard assay, which is commonly utilized to study the MMPs and MMPs inhibitors, which is gelatinizomography, for which we employed a cell culture of THP1 cells treated with the PMA to induce the differentiation as well as MMP's expression. And later, the cell culture was treated with the escalating concentrations of DTG with vehicle-treated controls, as well as doxycycline was utilized as a control because it is the only known US FDA approved as a broad-spectrum MMP inhibitor. So what we here noted in initial experiment, which we perform at least 5 to 6 times to confirm this outcome, that DTG significantly inhibits the relative activity of MMP2 and 9 compared to the controls. So once we confirm that MMP or DTG inhibits the gelatinases activity, which are MMP2 and 9, later we perform the fluorometric substrate assay to determine the effect of DTG on 10 different MMPs, which are known to be expressed or play a role in embryonic development, and were also representative of each subfamily of MMPs. And here we noted, interestingly, that DTG inhibits all the MMPs, however, at different extent or different degree. And then we also calculated the IC50 values from this experiment. And what we noted that the DTG has a stronger inhibitory effect on MMP 2, 8, 9, and 14. So this initial colorimetric as well as the zymography experiment helped us to understand that the DTG is a broad spectrum inhibitor rather being inhibition of the particular MMPs. Later, we wanted to understand the mechanism through which it inhibits the MMP's activities. So again, we performed the computational docking experiment using the Schrodinger software. So we wanted to know whether it inhibits the activities by interacting with catalytic domain or non-catalytic structural interactions. So we again employed 5 MMPs on which we know that DTG has a stronger inhibitory profile. Here, representative MMP2 structure is shown, catalytic domain with the zinc at the catalytic domain. And what we noted was the DTG forms a strong metal coordination with the zinc at catalytic domain of all the MMPs tested with the high binding energy. So these experiments helped us to conclude that the DTG is a broad spectrum inhibitor and it inhibits the activities of MMPs by chelating zinc at catalytic domain. I didn't show the data here, but we also performed this experiment with other integrase inhibitors as well, and we did see the similar chemical abilities associated with other integrase inhibitors for inhibiting the MMP's activities. But later we wanted to understand whether the DTG crosses the placental barrier, reaches to fetal central nervous system, and if it has any effect on MMPs or other biological activities. So we utilized the rodent model, which is C3HHJ mouse model, and these animals were administered with the DTG dolutegravir orally at supratherapeutic dosage, starting at the plug detection, which was gestation day 0.5, up to the birth of pups, and plasma from the dams was collected during the pre and postnatal period. As well as from few dams, the fetal tissues were obtained at gestation day 16.5, so we can determine the embryo brain exposure to the drug as well as effect on MMPs. So in the panel B, you can clearly see that we were able to achieve almost 10 times of the DTGC max levels known in the human plasma at therapeutic dosage. And these levels start to decline after the birth of pups at the postnatal period, obviously because we stopped the administration at the birth of pups. What important observation for us was that almost 700 nanogram of DTG we noted in the fetal central nervous system at gestation day 16.5, which indicated that embryo brains are getting exposed to this dolutegravir throughout the gestation period. So now we wanted to know the amount of DTG reaching to the fetal central nervous system has any effect on MMP's activities. The first question was whether to study individual MMP's activities or study the broad spectrum. How should we approach that? So obviously we know that the DTG is broad spectrum inhibitor, so that was the easiest way to do that. And other complexities of MMPs, particularly when it comes to in vivo studies, tissue in vivo studies, it's very complex about the MMP's expression, as well as there are overlapping functions of MMPs and codependent activation or functional properties. So we decided to determine the overall broad spectrum MMP activity assessment. So whole brain tissues were isolated from the fetal brains and processed for the MMP's activities from total protein. And we observed that the DTG significantly inhibits the broad spectrum MMP activities in embryo central nervous system compared to vehicle exposed controls. Later, we again performed this study. However, this time we perform with the therapeutic equivalent dosage. And in this experiment as well, we noted that, which is panel E, DTG significantly inhibits the MMP's activities in fetal central nervous system compared to the vehicle exposed controls. So now once we know that the DTG reaches to the fetal central nervous system and has some pathological effect or inhibitory effect on MMPs, does it cause any deficits in postnatal neurodevelopment? So again, we performed the similar study design where the pregnant dams were exposed to dolutegravir starting from the gestation day 0.5 up to the birth of pups. The drug was administered every single day, and vehicle-treated animals were used as controls. And adolescent pups aged between 28 to 36 days old were their biological assessments were performed utilizing the magnetic resonance spectroscopy as well as the transcriptomic assays. The assays were particularly intentionally performed like 8 days or 10 days later from their weaning from mother, just to avoid any effect from the stress associated with the separation. So the first, these pups were scanned utilizing the diffusion tensor imaging to determine the microstructural changes, and fractional anisotropy or FA values were calculated at 6 different regions of the central nervous system of these pups. And in all the 6 different regions, we saw that there is a significant decrease in FA values in DTG exposed pups. So as FA values actually are the indicators of anisotropic movement of the water molecules along the axons. Decrease in the anisotropic movement indicated the axonal or myelin deficits or injury. Later, same animals were scanned utilizing the magnetic resonance spectroscopy, and several metabolites were measured. Internal controls total creatine were similar between both the groups. But N-acetyl aspartate, which is the most concentrated metabolite of the central nervous system and the indicator of neuronal health or numbers or their integrity, was significantly reduced in the central nervous system of the pups who were exposed to DTG during the gestation period. So which indicated again the neuronal or synaptic injury. In the same in the same animals, total choline levels were also increased in the hippocampus, indicating the neuroinflammation. So the initial bioimaging modalities indicated the neuronal injury and neuroinflammation in the central nervous system of pups during the development following gestational exposure. However, obviously, we wanted to do the biological validations. So from the same animals, hippocampus regions were specifically isolated, and total RNA transcriptomic assessments were completed on total RNA, on which we observed that the around almost 2,594 genes were significantly differentially expressed in the DTG exposed pups. And when we performed further assessment for the enriched pathways, we noted that the majority of these dysregulated genes were associated with pathological pathways, which were linked to neuronal or synaptic toxicity or health, neuroinflammation, infiltration of immunocytes, or aberrant biosynthesis of extracellular matrix. So again, transcriptomic assessment validated or confirmed what we noted with the bioimaging studies. So one more point actually needs to be made here is the DTG administration to the mother was stopped at the birth of pups. So there is no DTG available in the central nervous system physically of the central nervous system of these pups. So whatever the adverse events we are observing here are due to the exposure to the drug during the gestation period. So now once we noted that yes, there could be impact on postnatal neurodevelopment of these pups following gestational exposure, whether we wanted to know whether it reflects into the behavioral impairments. So in the beginning, obviously, we didn't want to be restrict ourselves in determining what effect they could cause. So we perform a battery of tests with the help of our collaborator, Dr. Samuelson, who is the behavioral expert. And we perform all the studies kind of blinded manner, where like one group performed the animal study, one group performed the behavioral assessments, and one group performed the analysis. So we could have the higher degree of confidence in our assessment. So initially we performed the battery of tests, which included null object recognition, open field assessment, Y-maze, or gait analysis. Again, the same study design was utilized, same animal model where the animals were administered, pregnant animals were administered starting from gestation to 0.5 up to the birth. And the pups at the age between the 28 to 36 or 40 went through the behavioral assessment. So during the first Y-maze pre-exploration assessment, which is usually utilized to determine the spatial learning and memory. So, like surprisingly, what we noted, there is an increase in the spontaneous alterations in the pups who were exposed to DTG. So it does not really mean that the DTG actually improving the memory of these pups. I wish that was true, and I would be rich by then if I show like if drug is improving the memory. But that's not the case here. The Y maze also helps to understand the exploration behavior. And what we believe that here is what we are observing is the hyperactivation and exploration in the DTG exposed pups. And we also perform more studies which also help to confirm this observation, which I will present subsequently. We also later we perform the null object test to determine the recognition memory. So usually rodents have it's a natural tendency to prefer novelty, whether it's location, object, or anything. So in this test, usually the animals are trained during the training trial. They are provided the objects 2, 3 times. And during the actual test, one object which they are used to is provided, and one new object is also placed during the test. And here what we calculate is the recognition index, which basically determine the pup's preference for the null object. However, we saw that there was a decrease in the recognition index in the DTG exposed pups compared to the controls, indicating recognition memory deficits following DTG exposure. Later, we also perform open field tests to determine any effect on anxiety or depression-like behavior. And usually the mice with anxiety prefer to stay in the peripheral zone. They do not like to explore the central area, which is much more brighter and open. However, in this study, what we noted was DTG exposed pups were have higher speed, mean speed or maximum speed. They were exploring more in the center zone, or they were covering more distance overall compared to the vehicle-treated controls. Again, helped us to validate the same observation what we did with the Y-maze, that DTG exposed pups show the hyperactivation or hyperexploration behavior following gestational exposure. Once we noted that there is a higher activity or locomotion, then we wanted to perform the gait analysis for the motor functions and see if there are any differences. We did not really find any significant differences, but what we noted in this experiment was the increase in the body velocity of these DTG exposed animals, which also again at least helped us to understand that yes, these mice like to explore more or have more movement. So at this moment, like we are basically performing more experiments which are sensitive to the gait analysis, such as the voluntary wheel running, rotor rod, and then hopefully soon we'll have more or clearer picture about how DTG exposure during gestation period, or as well as other integrase inhibitors, affects overall neurodevelopment in terms of the functional outcomes. We are also performing the study, like initial group of studies will perform around day 30 or 40, but we are also doing the same assessment around day 90 and 100 to determine if these effects last uh, a longer period following gestational exposure. So obviously, till now, like we are the basic science group, we are trying to understand the effects in the mice. But like last year, we formed the collaboration with the Dr. Cassidy from Mayo Clinic, who is a PI on the multiple studies in Botswana, studying the functional outcomes as well as some imaging outcomes on the children who are exposed to antiretroviral drugs during gestation period. And we found the objective behind this collaboration was twofold through the exchange of scientific findings. First, to overcome the translational limitations associated with the behavioral observations noted in the animal models. And the second, to overcome the limitation with the observational studies performed in the humans by determining the biological or underlying mechanisms for cross-validated behavioral outcomes. Obviously, there is no support, or no one has claimed that there is a direct association between the rodent outcomes with the human behavioral outcomes, but we need to start somewhere and see if we can get something out of it. So this collaboration is ongoing, and hopefully we'll have some interesting outcome through this collaboration for the field in coming years. So in addition to the animals, around 2, 3 years ago, we got interested in utilizing or adding one more experimental model in our laboratory to determine the effects of HIV, antiretrovirals, or substance use co-morbidities on neurodevelopment, which were brain organoids. Actually, we started to work on brain organoids even a year or 2 years before it became a high commodity for the biomedical sciences or in general research. The reason we were interested in the brain organoids because honestly, they are the perfect model to study the neurodevelopment. They recapitulate or they reflect the initial 6 or 7 months of neurodevelopmental process in terms of genetic relevance with the human brain development rather than the adult neurobiology. I do not want, I know that these models are very widely utilized to study the adult brain, infectious models, or neurodegeneration. So I don't really want to make any claims or make anyone angry about that one, but I do strongly believe they rather reflect better neurodevelopmental biology than the adult brain. And subsequently, I'll talk about why also another reasons why we decided to use brain organoid models. So we all know that there are several experimental models which are commonly utilized to study the neurodevelopment. Each one has their own advantages, disadvantages. I'll not really get into the details of each of them, but like the monolayer cell cultures and rodent models are very widely utilized in the research field. And both of these independently or collectively lack certain aspects, whether in terms of the physical or genetic relevance, structural organizations, multicellular environment, which basically allows the cell-cell or cell-matrix interactions. However, in last 1 decade, during last 13 to 14 years, significant advances made to develop the complex and the more physiologically relevant brain organoids. So the brain organoids are nothing but self-assembled 3D tissues who closely recapitulate, or which closely recapitulates certain aspects of neurodevelopment in terms of multicellular environment, structural organization, genetic relevance. And such a multicellular environment is also critical because it provides that cell to cell or cell matrix interaction, which is very critical to create that microenvironment that determines the healthy neurodevelopment. And till now, they have been widely utilized in developmental biology to study the underlying mechanisms to understand the normal brain health or disease modeling, study the effects of medications or substance use, or effects of viruses such as SARS-CoV-2, Zika virus, and they remain a good model to do the high throughput screening for drug discovery or determining the efficacy or toxicity effects. So when obviously there are multiple brain organoid models as well. So it comes to the point like you have to decide which model or brain organoid model is more relevant for your research question. And for us, obviously till now, a lot of brain models or brain organoids are generated using dual SMAD inhibitors, which are called as a guided using guided protocols. And generally, these organoids generate cells only from ectoderm lineages, which includes neurons, oligodendrocytes, astrocytes. However, they lack a one important cell population, which is microglia. As all of us know that microglia are not really a brain resident cell type. It comes from these are generated from the yolk sac progenitor cells, and they are basically immigrants to the central nervous system. So we needed a model that also has microglia. We didn't want it to add from outside. We wanted the microglia which are getting innately developed from the mesodermal lineages. So we decided to develop the non-guided, utilizing non-guided protocol or without utilizing any external patterning growth factors so that there would be development of neural as well as ectoderm progenitor cells and their subsequent differentiation into the like neurons, astrocytes, microglia, and oligodendrocytes. So we obtained the induced pluripotent stem cells, commercially confirmed their pluripotency with the NANOG and OCT4. And later, with the non-guided protocol, we confirm with morphologically that there is a formation of neuroectoderm by day 10, expansion of neuroepithelium by day 15, and cerebral organoids start to generate by, or the structure starts to generate by day 20 to day 25. Obviously, we perform a comprehensive molecular assessment of our organoids to understand their development. First, we determine what brain region is getting formed. So the very robust expression of FOXG1 as well as AMX1 confirm the dorsal forebrain being the major brain structure of our organoids. Afterwards, we confirm the progenitor cells for where we confirm the PAX6 and SOX2 being the neural progenitor cells starting from day 20, and then they persisted throughout day 60. TBR2 confirmed the presence of intermediate progenitor cells. HOPEX confirmed the outer radioglial cells, which also is very critical because outer radioglial cells are specific to higher primates, which are not present in the rodent models. And also presence of brachyury throughout the study design confirmed the presence of mesodermal progenitors. Afterwards, we confirm the timeline or temporal kinetics of the cell development, where we can see that the early neurons getting formed by day 20 and their number or abundance increases up to day 60. Mature neurons start to appear by day 40, so, and which was also parallel with the myelin binding protein, which was the oligodendrocytes getting formed by day 40, and their abundance increases over the period. Astrocytes similar as they come from the ectodermal lineages. During the later part of development, they start to appear with less numbers at day 40 and their number increases by day 60. And the IBN microglia population, which is also critical for us, they start to form as clusters during early development and start appearing around day 20 and their number increases over the period. Once we confirm the cellular composition of these organoids, we wanted to also confirm the cortical plate or cortical structural formation. So, for which we also confirm the formation of early neurons coming from the deep layer cortical plate, which was confirmed that they start to appear by day 20, whereas the late-born neurons from the superficial layers start to appear by day 40 and increasing number by day 60, which again confirm the cortical structural organization in our organoids. So once we confirm a successful development of the cerebral organoids and the all the cells being present in our organoids with the confirmation of temporal kinetics or structural organization, then we treated these organoids with DTG at therapeutic as well as supratherapeutic dosages to determine the effect on their growth. So obviously we started the treatment of these organoids at day 20. That's when we transfer these organoids through the differentiation media. So we can see that initial exposure period, which is similar to human brain development. And the exposure for the initial assessment, the exposure was performed, continuous exposure was performed for total 20 days or 40 days, which was like day 40 of culture or day 60 of culture. And at both time points, particularly day 40, we can see that the both concentrations significantly reduce the growth of these organoids. In controlled brain organoids, the size was around 1500 or 1400 micrometer, whereas it remained less than 1000 micrometer or around there up following the DTG treatment. At day 60, the similar pattern continued where DTG treatment at both concentrations significantly reduced the growth of brain organoids. Whereas in the controls, obviously, you can see that the brain organoids grew in 20 days from around 1500 to around 2000 micrometer, which showed the continuous growth of organoids in controls. However, with following DTG treatment, it completely reduced their growth rather than being a delayed uh, growth. So once we understood that the DTG affects the initial growth of these brain organoids or cerebral organoids, afterwards we wanted to understand which cellular population is getting affected. So we performed this cellular profiling using the spatial transcriptomic assessment. Here you can see that the H&E stain slides were utilized to perform the spatial transcriptomics, and even with the H&E images, you can clearly see the reduced size of the organoids. And so we identified these individual cell types using the canonical gene markers associated with each cell type using a loop browser. And their relative percentage expression is basically presented here in the pie chart. So in the panel C, what was significant or notable for was there was a significant decrease in the mature neuronal population following DTG exposure compared to the control. It was almost in the control sample, 60% of mature neurons were observed, whereas in the DTG samples, it reduced down to the 49.25%. And it also paralleled with the relative increase in the neuronal progenitor cells as well as early neurons in DTG exposed animals, confirming that during early cerebral organoids, DTG actually affecting the neuronal development. So later we wanted to confirm the underlying mechanisms, like how exactly DTG is having an impact on neuronal development and what are the mechanisms. So we performed spatial transcriptomics and tried to understand the effects, where we did the total transcriptomics analysis actually. Due to the smaller sizes of these organoids and limited size, obviously due to cost associated with the spatial transcriptomics, we were not able to do the cell-based assessment right now, but that's what we are looking for in the future. So in the beginning, like obviously, we performed the enrichment analysis using ShinyGo for the biological processes as well as cellular components. And what we noted was the DTG again having the effect on neuronal development as well as neuronal cellular components such as the axons, synapses, or dendritic spines. And afterwards, we performed the IP analysis to determine the potential pathways getting affected due to the DTG exposure. Obviously, we have more gene data, but I'm not going to present that one. But what pathways where we found to be affected were associated with the chromatin regulation, cell signaling development, extracellular matrix remodeling, or intracellular protein processing. So from the spatial transcriptomics, again, we knew that the DTG has a majorly impact on neuronal development by affecting multiple developmental cellular processes. Then we again perform another study and perform the proteomic assessment just to determine that what we see at the gene level, whether it persists at the protein level as well. And here you can clearly see that the different clusters of the protein expressions following DTG exposure compared to the controls in the panel C. We almost observed like 4,000 or 3,400 significantly differentially expressed proteins following FDR correction compared to the controls. And the PCA plots shows the clear differences between the both groups for the proteome abundance. Again, we try to look at the pathways which are getting affected. Here, what we noted was mitochondrial homeostasis, intracellular processing, and again, the extracellular matrix organization getting affected following DTG exposure. But obviously, this is a big data, a lot of data comes out, but we wanted to know, identify particular DTG-linked molecular biomarkers which remain consistent at the gene as well as protein level, because we know that the certain pathways would be affected only at the gene level, certain will be affected only at the protein levels, but we wanted to see which remain consistent in both the parameters, so which can be utilized at large scale in the future. So what we noted was 4 particular pathways, which included mitochondrial dysfunction, energy pathway sirtuin signaling, protein ubiquitation pathway, and extracellular matrix organization being consistent at both genomic as well as the proteomic level. And we identified 12 specific proteins or genes which were affected in this comparative assessment. Obviously, we need to perform a little bit more biological assessment, but through this comparative analysis, we were able to identify specific key biological markers which were getting affected by the DTG during the early growth or development of the brain organoids. And now we will be looking into their association with the neuronal development as well. Afterwards, obviously, for the rigor and reproducibility, we again perform this experiment. However, we perform this experiment in the presence of bictegravir, just to see if it remains, say, INSTI class effect. So initial morphological assessment, the observations remain similar between the DTG and bictegravir, where both significantly reduce the overall growth of the organoids. And when we perform the proteomic assessment, what was interesting was, like, obviously there are clear differences between the 3 groups, but the proteomic abundance or the shift was different between the DTG versus control, compared to bictegravir versus control. So that was very interesting for us. Like bictegravir having like even though it is affecting the growth of organoids, it's having a different impact on the proteins. So we performed the correlation assessment and determined that uh, there are 1145 proteins which are similar dysregulated proteins similar between both treatment groups. However, there remained 2000 proteins remained on specific to DTG, whereas around 1000 proteins dysregulated proteins remain specific to bictegravir. So we did the further lot of assessment to determine what are the specifics to the dolutegravir pathways, what are specifics to the bictegravir. Obviously, even in the heat map, I know there are so many proteins and all that, but it's just visibly you can clearly see that the how DTG and BIC having a different impact on the dysregulation or the proteome of these proteins of these organoids. And when we did the further pathway enrichment analysis of what we observed, and which is very interesting for us for this study, was DTG having a effect on proteins which were associated with the mitochondrial homeostasis. It was very specific to the dolutegravir, like such as the pathways getting affected were mitochondrial ATP synthesis, aerobic electron transport chain, again, oxidative phosphorylation, and many more, which were only specific to DTG. And we have seen this again and again with several experiments of DTG DTG having an impact on mitochondrial homeostasis, so which we did not observe with the bictegravir. Rather, other pathways associated or other proteins and genes associated with the pathways such as the epigenetic regulation, extracellular matrix organization, or intracellular protein processing were remained similar between DTG and BIC treatment. So this initial study, what we concluded from this experiment was the adverse effect of both DTG and BIC remains same on these early growth of brain organoids, concluding that the adverse effects remain same among INSTIs. However, they could induce these effects through unique mechanisms. So now, obviously, we need to do more biological assessment to confirm that the how DTG has having a particular impact on neurodevelopment through mitochondrial deficits. In addition to the drug treatments, we wanted to understand how HIV affects the overall neurodevelopment. Like I understand that due to the antiretroviral drugs, there is no transmission of HIV is less than 1%. But due to the lack of models, till today, we don't really know if HIV infection happens during the early brain development in the microglia. How does it affect the microglial phenotypes? How does it affect their functions, which are very critical during the neurodevelopment? I mean, we know whenever there are deficits in microglial development, it could cause autism, schizophrenia, and many other functional deficits. So how does it affect the microglia, their interaction with the other neuronals, other cell population, because they are very critical for synaptic monitoring, pruning, axonal pathfinding, neurogenesis, precursor cell population maintenance, BBB formation. I mean, there are many more functions during the CNS development. So how exactly it could affect? So brain organoids remain a very critical model to understand that. And I'm very excited about this work. So first, obviously, we wanted to know the CD4 expression on the IBA1, which we confirmed that the CD4 expression remains dominant on our IBA1 population or microglia population. Later, we infected these organoids with the cell-free HIV, which is the HIV-1 EDA, CCR5 trophic virus. And we confirmed the productive HIV infection in our organoids almost 25 to 35 days after the HIV exposure. And we performed this experiment at least 2 to 3 times, and which was very positive for us that having this microglia getting infected during the early neurodevelopment. Now, obviously, we are performing the single cell assessment as well as single cell proteomics, so we can get better understanding about how microglia are getting affected due to the HIV infection and their interaction with other cellular population during the development. Obviously, we are doing those assessment right now. Hopefully, we'll have more data in a year. But what we noted was due to the HIV infection, there was a decrease in CD11B positive microglia population following HIV infection, which is obviously expected, but at least we see those expectations coming to reality that HIV infection can affect the microglia population during the development. Obviously, I have a time limitation, so I'll not get into those. But we, as I said, like we are also interested into developing the therapeutic intervention strategies, which have more of a translational relevance. So I'll quickly talk about this one and then I'll stop. So what we think that the long-acting formulations with through their pharmacological properties actually be beneficial to improve the safety of these antiretroviral drugs during pregnancy. So just to give a simple example, like systemic drug concentrations or half-life of orally administered drug depends on the elimination half-life. However, for long acting, it depends on the absorption rate from the injection site depot or cellular depot, and which leads to have longer half-life, dosing intervals, maintaining the drug levels as lower Cmax, as well as comparatively lower total amount of drug administration during equivalent duration. So for an example, if CAB is given every day, it will be 30 milligram per day, and during 2 months, it will be total around 17 to 1800 milligram of administration. However, bi-monthly CAB administration comes around only 600 milligrams, so which is almost threefold lower drug administration during the similar duration. So based on this, we hypothesize that the injectable long-acting, obviously we have been working on DTG, so that's what we utilize as an example to test our hypothesis that nanoformulated can provide improved safety by reducing drug fetal exposures compared to orally administered drugs. We performed this experiment in collaboration with the Dr. Gendelman and Dr. Idagwa from our department, who are very well known in the field of long-acting formulations. And after characterization of this study, we perform an animal experiment where animals were exposed to orally administered with the therapeutic equivalent DTG dosage, as well as long-acting formulations were administered either single dosage at the day of plug detection or one lower concentration of NDTG at gestation day 0.5 and one at 9.5. So this is what the interesting observation we noted here was the both long-acting formulations achieved almost therapeutic equivalent sustained levels of DTG in the maternal plasma. However, significant reduction of DTG exposure noted in the embryo brain as well as placenta, confirming the outcome that long-acting formulations can actually achieve the therapeutic levels in maternal blood but reduce the overall drug transfer to the embryo brains. We also performed the biological validations and noted that the long-acting formulations actually improve the mitochondrial functions as well as neuronal development or attenuate the effects of DTG by reducing the overall DTG exposure to the brain. So due to the time limitation, I think I'll stop there and I'll be happy if you have any questions. And obviously, I'm thankful to my lab members, collaborators, core facilities, as well as the funding support from NIH and other institutions. Thank you.
Thank you so much, Dr. Bade. That was a wonderful talk. As we are at the top of the hour, I'm thinking just a couple of questions. If you have any questions, please navigate to the bottom of the Zoom screen to the Q&A tab and you can type in your question. We already have one though. So since your hypothesis is fetal maternal and the CNS connection, how do your brain organoids address this question? How does this connection use NAMs?
NAMs as in
I'm not sure. Uh, would Sirapo like to expand upon that?
I mean, as I explained in my, like, just to give an answer, the brain organoids, as I explained during my presentation, actually, obviously, these brain organoids are highly closely resemble the early brain development in terms of the genetics, proteins, and everything as compared to the adult brains. So they are really good model to study the brain development. And in addition to that, they do possess the human microglia, which also allows us to study the HIV infection, latency formation, and all those formats during the brain development, which currently we do not really have any other model to study that.
Thank you. Next question. Do you have any data on the every 6 months intramuscular administration? Uh,
Uh,
go ahead.
Sorry, go ahead.
Currently in development in India.
Like as I said, like that's actually um data in terms of toxicology and from toxicology end on the formulations.
No.
Okay. I mean, like our lab is particularly focused on our lab is particularly focused on pharmacological and toxicological assessments or determining or developing therapeutic intervention strategies. So if the question is about the long-acting formulation development, that's not something we do. That's something our collaborators, Dr. Ganulman as well as Dr. Idagwa, work on it. So that's pharmacokinetics. We do not really do drug development. But if it's the question about the new formulations coming in market and whether we are have evaluated their toxicity or pharmacological profile in our models. No, we have not done that. We're actually trying to show how long actings actually could be a better option to improve the safety.
Thank you so much. So we are at time. I would like to quickly remind everyone of next month's Forefront of Virology webinar, which will be from Response to Readiness, Building One Health Molecular Diagnostic and Genomic Surveillance Capacity in the Caribbean, given by Dr. Christine Carrington. This will be held on August 27th at 9 a.m. Eastern Time. Thank you so much, Dr. Bade. It was wonderful to have you, and we look forward to seeing all of our attendees next time.
Thank you so much, Elizabeth. And I'm sorry, like there was not enough time, but anyone has any questions, please feel reach out to me through emails. I'll be very happy to answer any questions you have.
Thank you so much. Have a
Yep.
great day.
You too. Bye.